number 3 in a series of 5. And tonight we're going to move ahead in a description of this very elegant model of cancer treatment, which is called the metabolic model. And the title of tonight's talk is Glutathione, Mystery of Cancer. And we're going to talk about oxygen as a chemotherapy, believe it or not. So anyway, putting a plug in for my practice, you can schedule adjunctive cancer care by calling the number here, 202-237-7000 at extension 152. And you can ask for me or somebody on the Gant team, functional medicine team. All right, starting with a quote from Winston Churchill: men and women, humans, occasionally stumble over the truth, but most of them pick themselves up and hurry off as if nothing has happened. And November 3rd and 4th and 5th, if you want to come out and see the experts, see this exciting revolution of cancer treatment that's happening, and talk to us at Nyah. We'll have a booth and have discussions with various presenters. Come see us in Baltimore at this address, 692 Maritime Boulevard, but it's the Maritime Conference Center. And these are the times of the various dates that the conference will be happening. And there's a huge sense of excitement that's happening. There's a building, I don't know what you would call it, like a gestalt, an unconscious, collective conscious energy that's building, that is just so exciting, because this awful thing called cancer, think of the beautiful, lovely, wonderful people that it takes from us, who make such major contributions to their families, their communities, and the world. We really think we're about to make some major inroads into this, and we can help a lot of people. Okay, so first of all, let's review what we've talked about in the first 2 webinars. I presented the idea that I can prescribe or proscribe. Unfortunately, we don't do enough proscribing. Proscribe means to forbid, especially by law, but denounce, condemn, to outlaw. So I have in the past had a proscription pad, because there is a lot of health that comes from proscribing as opposed to prescribing. So I will proscribe sugar. Not only is our nation a nation of sugar addicts, the Western world, perhaps the whole world now, are sugar addicted to the tune of way over 100 pounds of sugar. That's those bags, those 1-pound bags of sugar, 100 pounds of sugar per person per year. And think about it, 100 pounds. But cancer cells are the ultimate sugar addicts, because it's the only thing that they can eat that gives them energy. The more undifferentiated they become, then the more they derive their energy exclusively from sugar. Okay? That's the important thing. And that process is called anaerobic glycolysis. I hate to use technical words. I don't want to put people to sleep with them, but there are a few words that are the words that are going to be thrown around. If you come to the conference in November, you'll hear this word thrown around a lot. But you can also just go on this website, NBC News, here's how sugar might fuel growth of cancer. That was in January 2016 in the media. And remember that starches, complex carbohydrates, you'll hear people say, oh, well, complex carbs are okay. No, they are rapidly converted by our digestive enzymes into sugar. So a baked potato, is a cup of sugar. So sugar is the food of cancer. The more undifferentiated, that is, the more the cancer cell has turned into a cancer cell and is no longer differentiated like normal cells, the more it depends on sugar exclusively for its energy. This is all review, and I'm sorry if you've listened to the first two webinars. We're going to move ahead with this model because the next major point tonight is how we can treat it as a prescription as opposed to proscribing sugar. So some interventions which lower blood sugar levels starve cancer cells from its addiction. Medication showing great promise. A lot of studies on glucophage. It has already shown to be a very effective agent. It's called a mystery in the journals. How does a diabetic medication mysteriously treats cancer? Well, it's not a mystery at all. By depleting sugar, cancer cells have nothing to eat because they can't. They depend on it so drastically and they can't metabolize fats or carbohydrates or protein for energy very easily, so they're totally dependent on sugar. So medication is out there. I think that there are better medications than glucophage, like Jardiance, which we'll talk a little bit about tonight. Insulin potentiation therapies are available at Nyah, IPT, and these therapies depend on making cancer cells extremely vulnerable by rapidly lowering blood sugar with insulin. Same way a diabetic would. Eliminating sugar, absolute sugar restriction, carbohydrate restriction, with a paleo diet. They call it a Paleolithic diet because before we were farmers, which we've only been farmers for 6,000 years, and then we've been herders for 12,000 years, so we get some sugar in milk products, but for the last 500,000 years and before that, we only had carbohydrates during the growing season. So it's called a paleo diet. Depending on your ethnicity, where your ancestors came from, the latitude on the planet, the growing season could be as short as 6 weeks to as long as, I don't know, 4 months. But for most of the year, there's no growing season, and the paleo diet includes no grains, no carbohydrates, no sugars, starches, mostly fish, seeds, and nuts was the diet. So the more we understand the biochemistry of life, it's very interesting. The better we get, the more we get into the science, the metabolism, we can test with great accuracy the personalized metabolism and genetics of each person. The more we begin to understand the shamanistic traditions of healing. And one of those is fasting. So if you had cancer 40,000 years ago, the shaman might say, you know what? It seems like fasting is a good way to detoxify And the last several people that had this wasting disease, I don't know what's wrong with you, but you seem to be wasting away to nothing. Why don't you do some fasting first, and then we'll put you in the sweat lodge and we'll cook you, and then we'll give you lots of water to clean you out. And you know, these shamanistic traditions are beginning to make sense now that we understand the biochemistry, the toxicology, the allergy, the immunology, and the genetics. Okay, other interventions which lower blood sugar levels. We want to lower blood sugar levels. We want to starve the cancer. Exercise, just burn it off with exercise. Now, I usually tell my patients don't overdo the exercise. Particularly if you're a little older, we may want to get some an EKG, do a stress test, an echo, get some sense of how your cardiovascular system is doing. But exercise is a good way to burn off excess carbohydrates. So the sodium glucose transporter inhibitors, SGLT1 inhibitors, like Jardiance, technically, theoretically, although we don't have the evidence yet the way we do with glucophage, should be much, much better drugs. But anyway, there are natural SGLT and 1 and 2, and also they inhibit the glucose transporters, which cancer cells, of course, since they're addicted to sugar, they have very high levels of glucose transporters. They got to get as much sugar in as they can, and this natural agent called organic apple root. I put a number here for you. We're just discovering how much to take based on tracking blood sugar levels with doing a glucometer testing in patients. How much of this do people have to take? The active ingredient is called fluoridazine, and you can look that up in Wikipedia if you want to read about it. And that's the phone number that you can call to get to buy some. All right, so we're still doing review here, and this is a cell, all the different components of the cell here, from the nucleus here, you know, and the ribosomes, which are here, and all the different parts, the endoplasmic reticulum, which is number 5 is here. A lot of metabolism happening there. But this guy right here is the key to what we describe as the issue in the metabolic approach to cancer. So that's this guy here. That they break open one, and you can see this inner membrane in here, but this is one from the outer membrane. It's called the mitochondria. Now the mitochondria is the energy plant. This is where electricity basically is generated, the fuel that runs life. So if you ask yourself, suppose you had a community, like you had a hospital here, and you had the stores here, and you had you know, the waste disposal place there, and you had, you know, different components to your community. And suppose your community was bombed all over. That is, something came in from the outside. In the case of the cell, that is viruses, radiation, and toxins that can come in and bomb the cell. But suppose we have a community of all these different parts in our community, and all the people live here in their apartments, you know, and so these bombs come in and they disable, like, you know, all the wars that are happening around the world. Or let's just suppose a 140-mile-per-hour wind shows up and just knocks down the buildings and knocks everything down. What is it that gets damaged that makes the most difference to a community. In Puerto Rico right now, what makes the most difference? Well, it's the inability to have electricity. Because without electricity, what happens? You can't have running water. You can't turn on any lights. The hospitals don't work. You can't do surgery, can't do treatment in hospitals. So then they have their own generators, of course. But look at all the things that go wrong. Eventually, you can't drive cars. How are you going to charge up your batteries without electricity? How are you going to transport food? So the mitochondria, it turns out, are perhaps, in many ways, the most important part of the cell. Certainly, they are the most important part from the metabolic approach. This approach to cancer as opposed to a genetic approach. Now, the mitochondria do have their own genes. They have about 33 or 35 genes of their own. But the genetic approach usually refers to the nucleus and damage to the genes, which is true. There is a well-defined genetic approach to cancer, but this elegant model of a metabolic approach, and the metabolism is about how food is burned and processed into the fuel that runs life, ATP. That's what we're talking about. Okay, so this is the mitochondrion here. And so what part of the power plant is the most vulnerable to being injured is the question. And the answer to that, like, it would be the same thing with a power plant. If a power plant was burned, or if a hurricane or tornado came in and started wiping everything out, or a flood came in, what part of the power plant would be the most vulnerable? Would it be the walls around the power plant here? No. Would it be the workers inside the power plant that are scurrying around and doing things? No. What would be the most important thing is exactly where the energy is converted. If it's a nuclear power plant, it would be where the radioactive fuel is converted into steam. If it's an electrical power plant with burning coal or fossil fuels, it would be the same idea. Where are the furnaces that convert the heat into electricity? That's the place. If those things get damaged, it's not only dangerous, they could blow up, but that's the high concentration of energy. And that's on the inner mitochondrial membrane right here. See, there's an outer membrane out here. That's the walls around the power plant. But this inner mitochondrial membrane is where the rubber meets the road, so to speak. And this can be damaged by viruses, toxins, and radiation. We talked about a major form of toxicity that nobody talks about much with cancer, and that last week. But I had to talk about it, especially since we're in the breast cancer month, where everybody's talking about breast cancer and wearing pink. Let's talk about how toxicities from addictive drugs, the self-imposed toxicities. That was webinar number 2. I had to get that out there because tobacco kills almost 500,000 Americans a year and 5 million worldwide. So everybody's, oh, it's heart disease, it's cancer. Well, behind the statistics are the toxins. See? And it's thought that 12%, I was very surprised to read the information about viruses. It's thought that 12% of the cancers in the world are caused by viruses. Isn't that amazing? And of course, radiation, X-rays. Okay. So this is another, this is the outer membrane here. See the outer membrane. Then there's a space in the mitochondria. Then they have this inner membrane. And in this inner membrane, sugar and fats and protein come in to be burned in what's called the Krebs cycle, or the citric acid cycle. And it just converts that energy, just like if you heat up water, it runs pistons, and the pistons then turn generators, and the generators then crank out electricity. So there's a conversion of kinetic energy and biochemical energy. So the citric acid cycle makes these intermediaries called NADH and FAD, and it even drives one of its Krebs cycle intermediaries, succinate. Now, all my patients know who this is, what this is, because every patient I see sees their Krebs cycle, and we evaluate it very, very carefully. And this then is made into fumarate that drives the cytochrome system number 2 and so forth. But anyway, right here is where the final product is made, ATP synthase. And ADP, adenosine diphosphate, is made into ATP. That's the jet fuel that runs life. And it has to do with moving hydrogen ions around, and here comes oxygen, and oxygen then is made into water. See, it explodes. It has this explosive energy of oxygen that then moves energy around on this inner membrane, and out comes ATP. That's the fuel. So all cells, whether they're undifferentiated or differentiated, whether they're cancer or normal, they derive 2 ATPs, only 2, from glycolysis. There's that word, anaerobic glycolysis again. And it's right here. Here's carbohydrates, sugar being digested. It comes into the cell and it generates pyruvate, and 2 ATPs are made. It's glycolysis. See? And the pyruvate then, along with these cofactor B vitamins and proteins, are broken down into amino acids. Okay, they come in, and fats are broken down. They come in, transported in to the mitochondria, and up to this point, there's only 2 ATPs being made. Now, the mitochondria can crank a little bit even without oxygen. 2 more ATPs can be made, possibly, if the mitochondria are not too damaged, in the citric acid cycle, and that's called substrate level phosphorylation. But basically, it's sugar to pyruvate. That's all that cancer can do. 2 ATPs. And all cells, regardless of who they are, have a certain energy requirement that is needed. If they fall below that level, they die. So, cancer cells can get energy from burning sugar, 2 ATPs. They might get a little more energy out of the citric acid cycle. But basically, that's it. They cannot use oxygen. See the oxygen right there? They can't use oxygen to get the rest of the energy. And what happens if you use oxygen in the mitochondria in the cytochrome system? You possibly can get 30 ATPs. It's called oxidative phosphorylation. You only have to learn 2 words. Anaerobic glycolysis, which is what all cells do. They get 2 ATPs there, maybe get a couple more, but only cells that use oxygen, there's oxygen down here, see? Only cells that can use oxygen, aerobic oxidative phosphorylation, only differentiated cells can do this. Cancer cells can't do it. So, how much carbohydrate do you need in a normal cell? Not much. How much protein? Not much. How much fat? You don't need much. So, a normal cell can get huge amounts of ATP from all these food components. Cancer cells are limited to 2 ATPs, basically. So, sugar becomes really important because their energy requirements are the same. Basically, all cells have a fairly narrow range of energy requirements. So cancer cells can only get it right there. So that's it, 2 ATPs. So they are totally dependent on sugar. So this is a diagram taken from Seyfried's great work, and Sheldon. This particular diagram was published in 2010 in Cancer as a Metabolic Disease, and this is the website for it. So this is being published now. It's in NIH, and there's a long tradition of revisiting certain approaches to various kinds of medical problems as new research comes in. So here's a happy cancer cell right there, a happy cell right there, see? And the energy requirement, we don't have to get involved with this, but for it's kilojoules per mole, and it's consistent whether you're a happy differentiated cell or you're a cancer cell, this guy here at the end of the line, say. And there's a gradual transition as you go from being happy to being totally dependent on sugar. So this graph here is the progression towards cancer over time. See? As a cell becomes gradually anaerobic, it makes this transition. So here's the percentage of ATP generation. Do you get it through anaerobic glycolysis, or do you get it from oxidative phosphorylation? A normal happy cell gets a small amount from, remember, 2 ATPs, maybe 2 more, a small amount from anaerobic glycolysis. A happy normal cell gets like 85–90% from oxidative phosphorylation. 30 ATPs from 1 sugar. But as the cell progresses towards cancer, it goes in this direction, little by little, see that here's a grade 1-2 PAP. This is a grade 3-4 PAP. This is a grade 5 PAP. This is a full-blown cervical cancer. See? Cells don't overnight go from 0 to 60 miles an hour without going through this transition. There's a gradual transition that happens. And as they go through this, little by little, they depend more and more on anaerobic glycolysis and sugar, and little by little, they depend less and less on oxidative phosphorylation until they only live on sugar and they do not use oxygen at all. So we basically tell these addicted cells, listen, you heroin addict, we're taking away your heroin. And it's going to make you very, very unhappy, but you have a choice. You can rehabilitate yourself and go back to being a normal, happy cell again. And we think that we can do that by detoxification, just like we detoxify addicts in a hospital-based system that are terrible addicts. We want to bring them back, rehabilitate them, and let them be normal members of community, be productive members of community, of a community. So a differentiated heart cell joins the heart cell community. A differentiated skin cell joins the skin cell community, and so forth. And they are good citizens. But a cancer cell is not a good citizen. A cancer cell is, first of all, an addict. And what do addicts do? They rape, pillage, kill, whatever. So they just hurt people. They can hurt society. So we want to rehabilitate the addict. If you're not rehabilitatable, then you die. So this is the message we give serious addicts. If you stay an addict, then you die. It's as simple as that. So you've got to be rehabilitated to come back and be a good citizen again. All right, so now we're going to go into some new stuff. That's the review. So there are 3 sources of injury that can cause cancer. So I had 2 patients last week with viral-caused cancer. And so the human papilloma virus is a big one, hepatitis B virus, herpes virus, and Epstein-Barr virus. These are viruses well known to cause cancer, perhaps due to damage to tumor suppressor genes. That's been one theory. But all infections cause oxidative injury. But the tumor suppressive genes could be, and I'm not saying there isn't a genetic model for cancer. Of course there is. But this metabolic model has been ignored, been given short shrift over the decades, and it needs to be looked at because there is a very elegant, keep it simple, sweetie kiss principle approach to cancer that needs to be looked at and considered. So my basic message to everybody is that whatever you do, whether you're going to do chemotherapy, radiation, surgery, great. Going to take an alternative approach, great. Whatever, you have a right to choose the kind of treatment you want. You're free. We believe in the sovereignty of the individual, the free choice in this country. We still have it, some of it, and you have the freedom to make your choice. So whatever you do, just make sure you pay attention to this metabolic model, because you can do it simultaneously. You can do it adjunctively, and whatever it is you do, it'll work. It just has to work better. It can't not work better. Okay, so there's oxidative stress from infections. There's oxidative stress from radiation. Obviously, that's why you're told be careful about getting too many X-rays, and sunlight protect yourself from sunlight, especially if you've got a history of squamous cell or melanoma in your family. Radiation. And then oxidative stress from toxins, which in my opinion is the big one. And we're talking about heavy metals. So we're going to get into this in the 4th webinar, detoxification, heavy metals. Arsenic, mercury, lead, and so forth. Pollutants. How do we get this garbage out of our body? Well, one thing's obvious. We're 70% water. So we have a river running through us, basically. So you put clean water in the front end. And then you want to excrete it any way you can. Urine, stool, sweat, run it through and clean out these toxins by putting clean water in the beginning. And then the body will prepare toxins that are not water soluble. Basically, our liver detoxifies by making toxins water soluble. See? And then all this water runs through us and out they come. So the oxidative stress, that is, oxygen is a very explosive fuel, and it's like at a power plant. If you're going to burn fuel, if you're going to use highly radioactive explosive fuel, and you don't keep it under control, you don't modulate it and keep it in a nice, even mid-range, the Goldilocks theory, not too hot, not too cold, you want to keep it just right. So the body burns toxins, but if you've got more toxins than usual, it turns up the oxidative heat. It starts burning toxins faster, and when it does that, you can overwhelm your normal protective antioxidants like glutathione and enzymes like superoxide dismutase. We test for these genetically. See, the genetic model's still very good, and we want to make sure that people have enough of their natural antioxidants, because if they're loaded with toxicity or infections, their oxidative stress can go very high. Well, like we talked about, where's the injury of viruses? Where's the injury from radiation, but especially from toxins? If you bombard your community, where is that bombardment? Where's that injury going to have the most impact? And it's the energy plant, the electricity plant. Because without electricity, everything comes to a stop. Can't get clean water. What would we do? Your cell phones would run out of electricity. Can't communicate with anybody. Can't communicate, can't have water, can't transport stuff. I mean, it's just, it's awful. So I hope you've donated money to Puerto Rico. I donated some money, and then when Puerto Rico happened, I was like, oh my God, I just open up the pocketbook, because man, these poor people going through no electricity, not going to have any for 3 months or something. Give me a break. So this is where the damage happens to that inner mitochondrial membrane, causing cells to lose their ability to use oxygen. They can't burn that fuel efficiently anymore, and they're then rendered into anaerobic. So glycolysis, they can only get a little bit of energy anaerobically. And so they've got to have the same amount of energy as a normal cell, so they've got to use huge amounts of sugar in order to do that. All right. So this is an article on the NIH website, Traverso et al. This is the most recent one I could find. There's probably more recent ones that talk about glutathione, the body's main antioxidant. It's connected to so many levels of metabolism, but it serves two main effects in terms of the metabolism in the body, which is to control the oxidative activity, to keep it in the middle just right, not too hot, not too cold. And glutathione is the main conjugator of toxins. So what does the liver do? Phase one, it burns the toxins, makes them ready to be reactive. And then phase two, it conjugates them to something to make them water soluble. So glutathione is one of the main conjugators of toxins. It serves two main roles metabolically: antioxidant and a conjugator. So it controls the burn, but if the burn happens, then glutathione is there to conjugate and make the toxin more water soluble. We're 70% water, and the toxins float out. So role of glutathione in cancer progression and chemo resistance. So maybe that's another word, but when people use chemotherapy for cancer, why does it stop working? Did you ever ask yourself that? How does the cancer cell adapt to it so that the chemotherapy stops working? Okay, so we're going to explain some of that here. So let me first start with this quote from this article. Oxidative stress, so the overburning of toxins or infections, white blood cells create oxidative stress to burn germs, has long been implicated in cancer development and progression, suggesting that antioxidants may provide protection from cancer. Well, of course we know that, right? Because oxidative stress damages this inner mitochondrial membrane. And if you damage it enough, you can no longer use oxygen. Right? Simple. All right, so it prevents cancer. On the other hand, pro-oxidant therapies, including radiotherapy, ionizing radiation, and chemotherapeutic agents, chemotherapy, are widely used based on the rationale, it's good rationale, that further oxidative stimulus added to the constitutive oxidative stress in tumor cells should, in fact, cause the collapse of the antioxidant systems, leading to cell death. And it does. Chemotherapy works. Radiotherapy works. See, you have a cell that can't deal with oxygen, so you're going to increase its oxidative stress, radiation, chemotherapy. And since it can't use oxygen, it falls apart, and it does. Well, however, there's always a however, right? This is a continuation of the quote. This latter approach, the chemotherapy and radiotherapy, has provided unsatisfactory results because many primary tumors express antioxidant enzymes at very high levels, leading to resistance of cancer cells to drug doses and radiotherapy. So here you come in, here's a cancer cell, it can't deal with oxygen, we're going to hit it with chemotherapy, radiotherapy. It works for a while, often works. We try to now to attune the chemotherapy, radiotherapy to the type of vulnerability genetically that the cancer cell might have in some way. So oncologists are getting very good at specializing the kind of chemotherapy to what the cell is likely to be most damaged by. But cancer cells are alive. What does life do? Remember Jurassic Park? Ever see Jurassic Park, where they were breeding dinosaurs? And they would breed all male or all female dinosaurs because they don't want them to reproduce. And the biologist that came to assess the situation got very upset because he said, wait a minute, this is life. Life will adapt to any kind of stress that it encounters, and you may have all females now, but somewhere along the line, one of these dinosaurs is going to flip into a male, or vice versa, and you're going to have a breeding problem that's going to get out of control, particularly if it's the Raptosaurus and the Tyrannosaurus. You're in big trouble, and that's exactly what happened. The cancer cell will fight back. The cancer cell is going to say, you know, I can't handle this oxygen. Even before you use the chemotherapy, the cancer cell is no longer using oxygen. What does oxygen do? It burns. It hurts. It puts stuff on fire. So the cancer cell basically says, I've got to do something about all this oxygen. I can't handle oxygen. I can't use it anymore. My inner mitochondrial membrane is shot. I can't get energy from food anymore aerobically. I can only get it anaerobically without oxygen through glycolysis. So oxygen is like pouring gasoline into a engine that no longer uses gasoline. It uses diesel fuel. The engine is not going to like it. See? What does it do with all this oxygen, with all this gasoline? It's got to dispose of it some way. So what does the cancer cell do? Well, the cancer cell makes huge amounts of glutathione. Huge amounts. An order of magnitude higher, because glutathione is an antioxidant. And the cancer cell can't use oxygen. Oxygen can injure oxidative stress. So cancer cells make huge levels of glutathione to protect themselves from oxygen. So along comes oxidative stress drugs and radiotherapy. So cancer cells, being living tissues, if you increase oxidative stress, what does the cancer cell do? It makes more glutathione. It protects itself. It digs in and says, oh yeah, I'm going to make more glutathione. So you use more chemotherapy. Cancer cell says, oh, I'll make more glutathione. I'll fight you at every turn, and eventually the chemotherapy doesn't work. You cannot out-oxidative stress cancer except for a few types, possibly the leukemias and lymphomas and so forth. But you're dealing with a living system that will adapt to anything you throw at it. So now you know one of the reasons why cancer cells lose their sensitivity to chemotherapy. Now, there are other reasons. Cancer cells will mutate. So there is a genetic basis to cancer as well. But I'll tell you, if we bring this metabolic approach and we combine it with the high-level genetic approaches that we're using now, and we get the genetic resistance of the host, of the individual, and metabolically adjust their genetic vulnerabilities, once we put all this together, see, this is why everybody's getting excited. The excitement levels are really building. All right, so this is called chemosensitization. And there are drugs like buthionine sulfoxamine, which block glutathione synthesis. Now, that seems logical, right? So you use chemotherapy, the cancer cells respond, they're sensitive to it, then they stop responding to it. So they stop responding to it because the cancer cells are adapting and making glutathione. So find a drug which blocks glutathione, and then the cancer cells will be sensitive again. Well, eventually, of course, what does a cancer cell figure out how to do? Eventually, it figures out how to get around that. It adapts, life adapts, and finds a new level of homeostasis to survive through. All right. So, Every patient I see just about, I get genetic testing with 23andMe, which is only, you can do the inexpensive version, $100, and then they won't give you the genes, they'll give you various aspects about your vulnerabilities, but they give you a link that for $30 we have an app that we convert that link into 300 or so of the most common, important, and modifiable SNPs, single nucleotide polymorphisms. Thank goodness they abbreviate as SNPs. Single nucleotide, that's quirky genes. The average person has 2 to 3 million quirky genes. Most of them are not common. They're not important. They don't do anything bad. And they're not modifiable. You can't do anything about it anyway. But there are several hundred at least. We'll probably find a lot more as time goes on of these SNPs that are common, important, and modifiable. And so we measure these 300 genes in every patient, and some of the genes we're looking for are the glutathione genes. Now remember, glutathione is expressed normally in a normal cell, but a cancer cell that can't do anything about oxygen generates huge levels of glutathione. And what does glutathione do? It's an antioxidant, and it's a detoxifier. It transfers the sulfur, which is one of the ways that we detoxify. We're going to talk in webinar number 4, we're going to talk about rehabilitating cancer cells, detoxifying and repairing the mitochondrial membrane so that the cancer cell can go back to being a normal cell again, possibly. Okay, so glutathione S transferase, GST, is the gene that synthesizes this enzyme, the enzyme which transfers the sulfur onto the toxin. Okay? All right. So this is the same quote from the same article that I just showed you. Polymorphisms, that is, quirky genes that decrease glutathione activity, are associated with a high risk of developing cancer. See, this is why we're very concerned about low glutathione levels. And if we find them, I test every patient for them. If we find them, we use glutathione oral spray to put more glutathione into the body. We test the 3 amino acids, glycine, glutamic acid, and cysteine, to see if one of those 3 amino acids is missing, and we supplement that amino acid. We provide glutathione IVs, probably the safest IV that there is, to just put it into the bloodstream, give the patient more glutathione so that they can detoxify, so that they can have more resistance to oxidative stress. Because what do toxins and oxidative stress do? They damage this inner mitochondrial membrane. And if you damage enough of these membranes, you can no longer make ATP, and you're on your way to cancer. So we use lots of glutathione. So polymorphisms that decrease glutathione activity are associated with a high risk of developing cancer. But an elevated expression of glutathione S transferase combined with high glutathione levels can increase the rate of conjugation and detoxification of chemotherapy agents, thus reducing their effectiveness. you see the logic of this? So if you have normal and high levels of glutathione, then you will not respond to radio or chemotherapy as well. So before you get cancer, it's the advantage is different from after you get cancer. If cancer happens, you're better off having low glutathione activity. See? Because that means even your normal cells can't make glutathione. And the cancer cells are depleted of it. All right? They can't protect themselves from oxygen anymore. So why are glutathione levels many times higher in cancer cells? Well, they're living things. They want to adapt to life. Oxygen is poisonous to cancer cells. Poisonous. I talked to a patient today, I hope she's here, that I couldn't figure out why she had cancer, metastatic cancer, for over a decade, maybe 2 decades. And she would go in her hydrogen peroxide hot tub 3 times a day and hit herself with all of this oxygen. And oxygen is poisonous to cancer cells. So she survived for I don't know, close to 20 years with metastatic cancer because she has blasted her cancer cells with this chemotherapy. Oxygen is chemotherapy for cancer. It dies. It does everything it can to protect itself from oxygen. Glutathione levels many times higher. See, here it is, progression towards cancer. See? And what do cancer cells, what do? They are totally anaerobic. Well, not totally, they are progressively more anaerobic as they become undifferentiated. What do normal cells do? They get almost all their energy from oxidative phosphorylation. So oxygen, it's not used anymore. It's not a fuel that can be used. And oxygen is bad. Oxygen burns stuff. Bacteria are mostly anaerobic. So they like to stay in the body. They don't want to leave the body because they have a nice, comfortable, relatively anaerobic environment. So bacteria make toxins that paralyze the intestines, and that's the only cause of constipation there is. Bacteria that are anaerobic and do not want to leave the body, they make toxins, paralyze the musculature of the intestines, like Clostridium, which makes the worst toxins that paralyze muscles. It causes tetanus. So these organisms make these toxins that do not allow the intestines to move. Primitive life is anaerobic. 80%, I'm just picking out a number, but I'm going to guess about 80% of Earth's history, when life has been here, has been anaerobic. Once oxygen showed up, it allowed for multicellular life. So the undifferentiated single-cell organisms fought back and found ways to live inside of multicellular creatures or hiding underneath dead trees and places where there's no oxygen. So oxygen is a chemotherapy. That's all, it's just chemotherapy. It's also very good for infections. Okay. So the more differentiated cells become, the more they become these stem cells, these primitive cancer cells, and the less oxygen they use, can't use it, and the more vulnerable they are to it. and the happier they are to not be in contact with oxygen. So they induce glutathione to not be poisoned by it. That's all. Everybody got it? Everybody got it? So what do we do about this? Well, if you've got sleep apnea or any kind of respiratory disease, this is a very dangerous condition for cancer. O2 levels are compromised. I mean, sleep apnea is a bad thing anyway. Germs and cancer cells love it when there's no oxygen around. So if you have periods where you stop breathing or whatever, and the oxygen levels drop, that's not good. Cancer cells and infections love this. They love low oxygen. So treating sleep apnea, very important. Fresh air. See, the more we understand life, the more we understand the science and the genetics and the toxicology and the immunology, the more we sound like shamans. What would a shaman say 30,000 years ago? Get out in the fresh air and breathe all that fresh air, because you're spending too much time inside the cave. And the oxygen, they don't know about oxygen, but they know that something's very bad about it, and the O2 levels can be depleted. Get outside and hyperventilate. Breathe deep. Ozone, hydrogen peroxide, IVs. Very helpful. Why? Because this is oxygen on steroids, basically. This is highly, highly charged oxygen. Now, your normal cells can handle this. The normal cells can handle oxygen. They've got glutathione. They've got all these antioxidant capacity to handle extra oxygen. Cancer cells don't. That's why they have to generate more glutathione. They just can't handle oxygen. So we use ozone, peroxide, hyperbaric oxygen chambers, same thing. Okay. Oxygen generators. You can hook them up either to a sleep apnea device or just wear the prongs, nasal prongs. Hydrogen peroxide baths, very inexpensive. And hot tubs. Never thought of that till recently with my dear patient there. But hydrogen peroxide baths, dumping a couple bottles of big bottles of hydrogen peroxide in the bath. Target sells them for $1. Inexpensive. High-dose vitamin C IVs. Vitamin C is an antioxidant, but when it's provided in high doses, it becomes a pro-oxidant. So that's one of the cheapest, most convenient ways to get into a state of oxidative stress that's safe, making it very hard on cancer cells. So we've known for years why high-dose vitamin C and peroxide IVs and ozone and so forth, hyperbaric oxygen. We've known for years that it works, but we weren't quite sure why. I wasn't anyway. I wasn't convinced. But you kind of like do what works, even if you don't know why it works. Now we're closing in on a model, on an understanding of what it is that works. So from the Traverso et al. Article, I won't bore you with any more quotes from it. Remember we said, however, the latter approach of using chemotherapy, radiotherapy, has provided unsatisfactory results because many primary tumors overexpress antioxidant enzymes, like glutathione, leading to resistance of cancer cells to drugs. That's the problem. All right, so here's the question. Cancer cells make very high levels of glutathione. They protect themselves from oxidative stress because they can't use oxygen, right? So what response should we take to this fact? See, this is the question. It's the whole point of tonight. This is the Socratelian dialogue here. This is the question. What should we do about this? What do we do about it? Do we take a pharmacological approach and increase the sensitization, the chemosensitization, by developing drugs which block glutathione synthesis? Well, that's logical, right? Drugs lose their effect because cancer cells fight back and make huge levels of glutathione. So we increase the drugs, they don't work anymore, so let's make the drugs more sensitive by blocking glutathione, right? I mean, it's logical. The metabolic approach is very different. The metabolic approach is what are cancer cells saying to us? They're living things, and what are they saying? They're saying they can't stand oxygen. That's the message. That's what they're communicating. So if they can't stand oxygen, why don't we hit the cancer cells with every form of oxygen possible? That's the answer. See? Or maybe some combination between these two. Block its ability to make glutathione with a drug, hit it with drugs that increase oxidative stress. All right, radiotherapy, all right. But make sure that you're doing everything you can to increase the oxygen through all those different technologies, because cancer cells can't stand oxygen. And we know they can't stand oxygen. We knew it from the first lecture here, because we talked about how cancer cells are anaerobic. So we didn't even need the research. Thinking out of the box, we would have said, oh, they can only get energy from anaerobic glycolysis. So probably they can't use oxygen anymore. So oxygen probably hurts them. I mean, we could have made that leap just by knowing the inability of cancer cells to use oxygen through oxidative phosphorylation, right? Now, it's very interesting to me how it would seem that conventional medicine has this idea that life is something very hostile and we have to damage it in some way. We have to block an enzyme. We have to hurt life in some way in order to control it. And at times, when you have something as damaging as cancer, sure. But from the integrative approach, life is an adaptive mechanism that responds to its genetic makeup and its environmental stressors to adapt in a way that survives. And if there's one thing you can say about cancer cells, man, they want to survive. I mean, they're addictive as hell to sugar, and they can't stand oxygen at all, but damn it, they're going to find a way to survive. And boy, they do. So we simply take away their resources. We starve them, don't give them sugar, get blood sugars down to in the 60s if possible, and poison them with a wonderful chemotherapy, which is so safe. I mean, how safe is oxygen? We breathe it. How many times a minute are we breathing oxygen? It's safe. It's amazing we need a medical license just to give it to people. So you poison it with oxygen, and you starve it by taking away its carbohydrates. I mean, could the model be any more elegant than that? So there's a problem with antioxidants. If you want to prevent cancer and increase apoptosis, that's another term that's used, that's cancer cells dying, increase the likelihood that the immune system and white blood cells see these huge amounts of oxygen. But they need to control. They could be damaged by oxidative stress too. So antioxidants prevent damage to the inner mitochondrial membrane. If you have infections, you have toxins, that oxidative stress can go up, and antioxidants of all kinds are very good for you because they prevent cancer cells from forming. But if you have cancer, so now the damage has happened, antioxidants may be very bad for you. Now, for my integrative medicine colleagues or people who have this integrative view, the idea of antioxidants being bad for you is just a terrible idea. It's a foreign notion. And I'm sorry to think out of the box here and I know it's a sacred cow of integrative medicine. Vitamin C, bad. Glutathione, bad. Berries have very high ORAC value, and they're too high in sugar anyway. Bad. Turmeric, green tea, antioxidant herbals, bad. And oncologists have been saying this for years, and I've resisted them because, the studies were, it prevents cancer, but you can basically, the saying in medicine is, when you do a study, you can torture the data long enough to make it talk. You can basically, as you torture the data that you get, you can kind of reach any conclusion you want. I mean, I'm sorry, but that's kind of a sacred cow too for a lot of people. And I believe there is integrity in science. There is still a lot of integrity, but you have to read the study to see how many flaws there are, how many presuppositions were made, and how they took the data and then reinterpreted or moved it around. You have to look at that to get an idea as to how much truth there is in scientific discovery. But there is a closer assessment of truth. But see, oncologists have been preaching for decades, look, we're trying to increase the oxidative stress. Our chemotherapies are doing that. So don't use antioxidants. And they will still say that to patients. Stop taking vitamins, stop taking vitamin C, stop taking antioxidants. And they have a point. They have a point. Why would you protect cancer cells that can't stand oxidative stress when the cancer cell is doing everything it can to make its antioxidant substances so that the cancer cell won't be damaged by oxygen? And here the chemotherapy is trying to increase oxidative stress to damage the cancer cells more. So now there can be a meeting of minds as the metabolic model, which is so elegant and so simple and so absolutely irrefutable. There can be a meeting of minds here. Okay, so there's the man, Winston Churchill. Men occasionally stumble over the truth, but most of the time they pick themselves up and hurry off as if nothing had happened. So that's the name of this cancer conference, Tripping Over the Truth. I think they derived it from the Winston Churchill quote. I don't know. November 3rd, come out and join us. Now it's not cheap to come and listen to the latest approaches to cancer, to go around to the booths and hear the latest information, look at the different products that are available and so forth. But check it out, and I hope to see many people there, and you'll hear many offshoots of this metabolic model happening that further consolidate this model that we're all so excited about. November 3rd, 4th, and 5th. And of course, shameless ad for my practice here, Nyah's number. That's one of the extensions you can call, or operator. Thank you for being here tonight. You take this time out of your life to listen to something that I think is so important. But we're all busy. We got so many obligations in life, and some people just care as much as you. So thank you for being here tonight.